Auf den Spuren der Schleimpilze. Ein Podcast für Neugierige.
Welcome to yet another episode of On the Trail of Slime Molds. It's great having you with me today. My name is Anna Bursac, and I've recently graduated from high school in Vienna, Austria, and I produce this podcast about slime molds as part of my final research project for graduation. Today's topic is the following question. How do slime molds reproduce? Spores. This is something they have in common with fungi, which also rely on spores for reproduction. These spores are incredibly small, typically about 7 to 12 micrometers in size. Once produced, the spores need to be dispersed. This can happen through wind, rain, or animals. Beetles and mites, for example, feed on slime mold spores. The spores pass through their digestive systems, survive the process, and are eventually excreted while still capable of germinating. Spores can remain viable for years, at times even decades. There has been the case of a 75-year-old spore, still capable of germination. Now let's return to spore dispersal. Wind is the most important and least energy-consuming means of transport for slime mold spores. The spores are also well adapted to this method of dispersal. They possess so-called surface ornamentation, meaning that their surfaces are often covered with tiny hair-like projections, spines, or wart-like structures. These features allow them to remain suspended in the air for longer periods, and, as a result, spores can travel longer distances. The primary function of spores, therefore, is the dispersal of slime mold. Eventually, they land somewhere, ideally on a suitable substrate. If temperature conditions are favorable and food is available, the spores germinate. Interestingly, isolated spores tend to germinate less successfully than spores that remain within a large spore mass. Meiosis taking place inside the spore is the beginning of germination. Meiosis is a special form of cell division that produces between 1 and 4 haploid germ cells. These germ cells are required for sexual reproduction. Once they have formed, they are released from the spore. Depending on the degree of moisture in their environment, slime molds can produce 2 different forms of germ cells. In very moist environments, they develop swarm cells known as myxoflagellates. These cells possess 2 flagella and can swim. When moisture levels are too low for swimming, slime molds instead produce amorphous myxamoeba. These cells move by crawling, much like ordinary amoebae. Particularly remarkable about these germ cells is the fact that they remain adaptable even after they have fully developed. They can transform from one form into the other. Myxoflagellates can become myxamoeba, and myxamoeba can become myxoflagellates. For this reason, the germ cells of slime molds are often collectively referred to as amoeboflagellates. At this stage of the life cycle, enormous amounts of germ cells are released because many spores germinate roughly at the same time. Naturally, environmental conditions do not always remain favorable. The habitat may become too dry or food resources may decline. When this happens, each germ cell forms a protective capsule around itself, since inside this capsule, it has much better chances of surviving adverse conditions. These dormant structures are known as microcysts, and these germ cells, they tend to remain close together, forming what is known as a pseudo-colony. Once conditions improve, the microcysts release active myxamoeba or myxoflagellates again. If 2 compatible germ cells successfully fuse, fertilization occurs, and a zygote is formed. The zygote then develops into a plasmodium through a series of synchronized mitotic cell divisions. Over time, the plasmodium becomes multinucleate. This is because its nuclei continue dividing repeatedly, but cytokinesis, the division of the cell itself, does not occur. As a result, the plasmodium remains a single giant cell containing many nuclei. Through continuous feeding, it grows larger and larger. In most cases, a plasmodium remains intact as one single cell. However, there is one stage in its life cycle during which division becomes much more likely: the formation of fruiting bodies, also known as fructifications. To initiate this process, the plasmodium migrates toward brighter and drier locations. The number of times a plasmodium can divide varies considerably and depends on its size as well as its capacity to produce fruiting bodies. The fruiting bodies of slime molds are extraordinarily diverse in form. Their function is to produce and contain the spores, whose dispersal we discussed earlier. As the outer surface of a fruiting body dries out, it eventually ruptures and releases the spores into the environment. What a fascinating and complex life cycle. And with that, we have reached the end of today's episode. Thank you very much for listening. I look forward to having you join me again for the next episode.
Sie haben gehört, auf den Spuren der Schleimpilze. Schön, dass Sie heute dabei waren. Bleiben Sie dran.